the benefit of all the participants from USA, family members of Dr. Shastry Garu, and my IIRR colleagues, I heartily welcome our Dr. Seshu Garu, who is very much associated with Dr. Shastry Garu. And we thought of organizing this lecture from first after completing our first Shastry Memorial Lecture. And this is in the sequence of second Shastry Memorial Lecture. And I'm very thankful that with small, without small, is readily agreed. And he was very much interested to deliver this lecture, sir. Thank you very much from all the IIRR colleagues for your immediately accepting it. And we are eager to, actually, we are having a program on 15th December. So that will be in the morning hours of in the Indian Standard Time. So where we wanted to telecast this, whatever we have recorded today, it will be telecasted on 15th, our regular program, Foundation Day program, which is there on December 15th. So we wanted to record it initially. Then same thing will be again telecasted with all the cooperators of this society, as well as the members of the DRR staff members. So thank you, sir. Once again, I welcome you for this lecture. And now the floor is open for you, for your lecture, sir. Once again.
Thank you, Dr. Kumar. Greetings to all my fellow crop scientists and to family members of my good old friend, Dr. Shastry. And I feel it a great privilege and pleasure, to deliver this memorial lecture in honor of the departed soul and a very close friend of mine and a very long time professional colleague, Padma Shri Dr. Sishta Venkata Seetharama Shastry. For my lecture, I chose the topic strategic for the networking for genetic enhancement at a global level. I chose this topic because of two reasons. One, that is a subject very close to the heart of Dr. Shastry. Second thing, my own involvement for several years at national, regional, and international levels of networking. National, the ICAR, I was there for 10 years, and in the early 70s, when it was established, I went there as a consultant. And of course, my long association with the International Network for Genetic Enhancement based at IIRR. And I also had the privilege of establishing a genetics network of fish at the World Fish Center. And so these are the reasons I thought I would be able to summarize my experiences to the benefit of my audience. And that's the reason for the subject. But before I proceed to my lecture, I would wish to say a few words about Dr. Shastry. Our association dates back to 1955, when I joined the Central Crisis Institute at Cuttack, and Dr. Shastry was already. And at that time, both of us were working on the Japonica indica hybridization scheme. And the focus of that project was to develop varieties which are fertilizer responsive. And many of you must have heard the name Masuri. It's one of those products of that. And after that, we had a overlap of 1 year. Studies in Wisconsin. And after he returned, he took up a job at ICAR-IIRR. And there he again pursued rice and focused on the rice cytology. Simultaneously at that time, I was working on the cytogenetics of interspecific hybrids of varietal. And thus again, we had opportunities to interact, particularly, when he was trying to find out the cytomorphological characterization of the Pachytian chromosomes. which until then was an uncharted territory. And Dr. Shastri took a bold venture to work on that with his colleagues. And because of a common interest, as I was involved in the cytogenetics of interspecific hybrids, we used to interact very frequently and even exchange materials and exchange visits. And after that, once again, our paths crossed when ICAR was established, and Dr. Shastri was heading the project, and I was heading the breeding program, and worked together for 10 years. It was really an exciting period in our professional careers because that period signaled the Green Revolution. It is absolutely, I would say, no, facilities did not exist. That Dr. Shastri, Dr. Freeman, and I, we used to share one room along with some important breeding materials. But however, the paucity of funds or lack of physical facilities, those have never dampened our enthusiasm because of the excitement and hope and promise of this new technology where we are to change just like tailoring the plant with a new physiomorphological architecture. And with that hope and the excellent leadership provided by Dr. Shastri, we worked in a good team, worked as a good team. In fact, a comment by then Deputy Chairman of the Planning Commission, which I think my IIRR colleagues can see if the visitors' books are still there. The comment says, very few staff members, but very large output. That I think is a big tribute to the and the work that we got involved in changing the plant type. And after 10 years or so in 1975, Dr. Shastri got an offer from the FAO, and so he had to leave ICAR, and I took charge of that for a few months before I got a call from IRRI to join their staff. At IRRI, I was the coordinator for at a global level for the international network, which initially was called IIR. At that time, Dr. Shastri, with a brief stint with FAO, he moved to IITA, and because rice is also an important crop, he took advantage, and then when he pursued as a director of research there, the program rice, we have established a regional unit in Nigeria for the West Africa, and that was located in IITA. That's how we continue to interact. And after retirement, I had a consulted on a couple of occasions. And when both of us retired, we frequently used to interact with each other. And I can say that even the latest of our interaction, I found that intensity of his interest in the subject and his sharp memory and brain, I was quite impressed with that, though no different from what it used to be during the active days in ICAR. And on the other side, on the personal side, I know that Dr. Shastri, well, he has been, his contributions have been well recognized and awarded. And I think all of you know about that and his contributions. But I would, from my experience, had my few impressions that one is that he is a scholarly eminence that really reflects on a combination of memory power. In fact, those traits worked as an excellent leverage for the leadership he provided to the ICAR and became pivotal for the success of ICAR. And he has, in my umpteen interactions, whether professional or not professional, I was so impressed with his quick grasp and then very critical analysis. And I can explain his missionary zeal with no room for any nonchalance at a time. And he has a very unique personality, and he doesn't fight shy to speak his mind, forthright, candid, and he wouldn't bring into play the art of diplomacy. And so I was quite impressed with that. On the personal side, I know that he is deep interest in the mythological stage plays and also, in games like Bridge. But what we and our family remember is about his culinary skills. When I was at Cornell University as a visiting professor, Dr. Shastry happened to visit and stay with us in Ithaca and prepared some excellent food. And my children, even today, they talk about the pizza that he made. And that's that Shastri. Let me now show few pictures, and I think these are of, I would say, historical importance. For the history of ICAR, they should be, I would say, more classical. Let me show the this is the flagship variety of ICAR, which was named as Jaya. And this was in the making, which we used to have Prodhanero trials. And this was developed using the traditional variety type 141. And what happened is at the level of this trial, at the Prodhanero trials, we had several sister lines giving similar performance. Then, because they were not showing any statistical significance in the normal analysis, however, we had to apply the Duncan multiple range test, and then we found one line IET 723, which has been very consistently performing above its sister lines and others in the All India trials. And that breeding line IET 723 has become ultimately the variety Jaya. And here, friend Heman Prasad, a fellow breeder, but unfortunately, at a very young age, he passed away. Next, again, of historical importance is the visit of Dr. Norman Borlaug. And here in the picture, you see him taking notes in the ICAR field trials. Dr. Shastri, Dr. Freeman, and myself were with him. Let me, at this point out about Dr. Freeman. He had a deep commitment, and then his work aptitude and then the interest in the Indian agriculture was very much evident from his contributions at that time, and that is very well complemented with the leadership of Dr. Shastri in bringing success to ICAR. This is, as I told you, in the earlier stage, we used to just share one room. At one time, we were in the Kirkan Center, the Sorghum Center, one room. Then we moved to the ARI of Agriculture University, and there again, a couple of rooms. And finally, a physical facility was provided. That is the old one. I think all around that building now you have so many buildings added. And that was inaugurated by the then President Fakhruddin Ali Ahmad. And also you can see here the Chief Minister then of Andhra Pradesh, Dr. Venkata Rao. This is yet another important because, when we started the program, we used to have the annual workshops, but then to get a preview of this to the state's representatives, we used to have the zonal coordinators, and we used to have the meetings with the zonal coordinators before we have the workshop. And this is first of those meetings where you can see Dr. Freeman presenting some notes. And This I thought the IIRR, if they do not have, should have in their collection, because the first of those interactive meetings. This was the farewell captain mission. He just left for Rome to take up that assignment in FAO. But as I told you before, after a year or so, he left for IITA. Now, with those few words about Dr. Shastry, let me start with my presentation. The strategic networking for global rice genetic advancement is my topic. Historically, there used to be very, very limited exchange of germplasm before the Second World War. But in 1950, the United States Department of Agriculture put together a wheat stem rust nursery. This was planted by the serious appearance of the wheat stem rust. And so that's the first time the materials have crossed national boundaries. After the international centers were formed, IRRI likewise sent a set of materials to find the reactions to the Pericularia oryzae pathogen, and likewise semi-test blend spring wheat yield nursery. This is just a preliminary to a structured networking. But coming to the structured networking, where you have cooperators sending their materials and exchanging and testing at national level, perhaps ICAR was the largest, and more than 100 research centers from different states of the country participated. Likewise, in case of the international level, the IRTP later we changed the name to INJUR to better reflect its activity. Now let me focus on the network. I will have three items. One is the concept, and the second is the impact, and the third is so what should be. Concept is, first of all, to promote genetic diversity, you need to have an access to wide range of germplasm. And again, access to hotspots, because the reason is you find a lot of insect parasites, and then pathogens keep attacking the rice crop. But in many cases, those are seasonal, that one season they may occur, one season may not. But there are hotspots where season after season those occur. And so for the national program scientists, it would be a big advantage that they can utilize those hotspots so that they do not waste time in finding the resistant varieties. And by pooling up the resources and pooling up the materials, while the time for developing a new line has been significantly reduced, the costs are also saved, and then there's no redundancy can be avoided, because each one can supplement their efforts. Ultimately, it is the acceleration of a transfer of technology to the farmers that is the ultimate goal. Then the network studies, when you get the data, some of them very suggestive of very special studies. So this network can be used as a platform or a springboard for those special studies, of which I'll give you some examples. Now, the INJUR itself, that is the network concept. INJUR was participated by more than 70 countries from Africa, Asia, Latin America, and Oceania. Now there is a very cultural diversity as well as diversity in political faiths in the countries that are participating in the network. More important for the breeders is the test sites represent a wide range of diversity in the agroecological conditions. So now you have the diversity of the agroecological conditions, and then you have a geographical diversity. Earlier, that rice was confined primarily to the semi-aquatic conditions, but over a period of time, it made forays into a wide range of ecosystems. That's why the agroecological diversity poses a great challenge for the breeders for the genetic improvement. However, the geographic an opportunity for cooperation. Now there are several countries participating, and they are not all at the same level of strength in terms of physical facilities or the scientific capability. But when you make a meaningful fusion and complement those strengths, then the collective strength would be definitely very formidable. With also many countries participating and with also many the research stations, more than 1,000 and thousands of rice scientists involved in it, India turned out to be the world's largest agricultural research network. For interaction, one is a joint site visit. When there is a joint site visit, scientists from different group of scientists, depending upon the subject and interest, visit certain nurseries in certain countries. And there they get to see how though others conduct the trials. And there they get to have interactions with the fellow scientists. And so this will be an excellent forum for communication and interaction. The other forums are, of course, we used to organize a series of workshops with focus on different subjects or different ecosystems. And then we have scientific advisory committee for the testing program. And then, of course, there are individual exchange of visits among scientists. So these are the various important. Now let me talk about the impact of this program. More than 1,100 varieties as of date were released in 74 countries across the world, and several entries in the breeding programs. Now, the multi-location testing provides an excellent opportunity for determining some items like which I listed, stability of performance, and biotype and pathogenic risk variation of the major insects and diseases, and physical factors like rice weather interactions. So that provides an opportunity because of the variability in those both biotic and abiotic stresses. And then, as I indicated before, for specific biotic and abiotic stresses, it gave an opportunity for identify the hotspots. And of course, important and to avoid genetic vulnerability where there is a crop that is grown uniformly. And so the diversity gives a genetic buffering so that the crop can withstand for a few years against these biotic and abiotic stresses. That genetic diversity was created, as you would see in the breeding programs of the various countries. Here is one example I'm giving here. The rain-fed lowland ecosystem is one of the very important ecosystems, particularly in South Asia. And here, a Bangladesh breeder has a choice to make. What we do is, since the individual factors contributing to the yield cannot be systematically quantified, the mean of the test sites yield trial would in way has a compounded effect on the various factors that cause that particular yield. Now, when you have a regression of the individual site performance for a given variety against the site mean, that shows the performance here. The picture which I'm showing is showing a linear response. Your attention on the two varieties. This is BR 51288 in Bangladesh. You would see that its performance was very good, more than 5 tons when the conditions are highly favorable. But when they are not, it is below 1 ton. Whereas on the other hand, this line 859.1 across different levels of the growing conditions, it showed some stability. It's not that you want that a variety to shoot the moon in 1 year and then go into the depths of the ocean the next year. So these varieties like this, would provide the stability, and this would not have been possible to get the information so quickly, because in a given year they would not have all these conditions. But across locations, you would see that within a short time they could see which is the variety under the adverse conditions, cultural conditions, can identify varieties that are more suitable. Similarly, the multi-location data has provided sites which show distinct variability in weather factors. And here in this picture, I'm showing the isoquants. These are the contours, two-dimensional contours, and solar radiation. And on the y-axis, the minimum temperature during the flowering and ripening. Now, if these are curves for equal predicted yields ranging from 3 tons to 6 tons. You see here at this combination, for example, when you look at a solar radiation availability of around 350, and the temperature was around 24 degrees during ripening, you have only 3 tons for yield. Now, even at that low solar radiation level, when the minimum temperature during the ripening phase has has 6 ton yield, these are the isoquant plots anyway. So that shows that the lower temperatures during the ripening phase and higher solar radiation would give the best yields, and that's why in the temperate country yields are quite higher significantly than in the tropical countries. In fact, with that lower temperature, the ripening phase gets extended, and during that process, it gives an opportunity for the plants to get more and more of the solar radiation. So that combination, which I put here in the microwatts per square centimeter in the range of 300 to 600, possible if you are operating by yourselves in a single station. And the importance of the network was very much illustrated by that. Now, another example which I'm giving distances. Here, you may not be clearly see whatever is written there, but let me tell you those bars in 3 groups. The one in the extreme left, East Asia, and South Asia. Now I'm showing here with a common set of varieties. That set was sent to these different countries in different regions. And you see in the common set how in South Asia, the percentage of susceptibilities, even the distinct differences, day and night differences between that. Whereas somewhat moderate in Southeast Asia, and that shows the opportunity it provides for selecting the location-specific the biotypes of the insects. So insects like they have very important in the sense they can cause damage both directly and indirectly, as you all know. Directly as feeding on the crop, indirectly they serve as vectors for a major virus known as grass-resistant virus, which can chew up the crop in no time. However, if you have controlled the vector, you would have controlled the virus. Where genetically, it has been possible that we could identify very highly resistant varieties for the insect pests. Similarly is the case with the pathogenic variation in case of the bacterial disease, the bacterial leaf blight. Here I'm showing you on the extreme right in South Asia, the percentage of resistant entries from the same common set. And then you would see that most of the entries resistant in Southeast Asia, where IRI is located, so they're all susceptible in India, Bangladesh, etc. That shows how much the location-specific focus on these things is so very important. Yeah. Now, continuing the impact, the nurse breeding research capabilities, and then the institution building, those are really, I would term it as a progress from dependency to interdependency. This is very well reflected by the fact that initially when we started the program in 1975-76 period, nearly 90% of the test entries were from ERI. And there are very little contributions from national programs, and most contributions from the national were from India. However, 10 years in progress with this network, the national capabilities have strengthened because of the participation in the network and participation in the workshops and joint site visits, and then, more importantly, their interactions with the fellow scientists across the world. The progress is now from dependency to interdependency, and 10 years after the participation, 75% of the entries are contributed by the national program and remaining from the international centers, mostly from ERI. Another thing is that in terms of the economic impact, we had a consultant, Dr. Evanson, a very famous economist from the Yale University. At that time, around 570 or so the entries from India were released as varieties. So they worked on that impact of those varieties, and with their calculation, he and his colleagues, with their calculation, they concluded that every injured variety contribute to $1 billion. Now, based on that, that was in 1992, where about 600 or 570 varieties were released, but now it is more than 1100. And then, of course, also the inflation is there. If you factor in those changes, then I estimated it to be $2.4 billion today, each variety is worth in adding to the world economy. Third and most important from the ERI point of view is that, while ERI is located in Philippines, which represents only one of the several ecosystems, or one of the rice environment, total environment, whereas the varieties, as I indicated in the earlier slides, those are developed in ERI, would not be of any avail to countries outside ERI when there is a pathogenic or biotype variation. And therefore, the INJR acted as an excellent outreach arm for implementing the IRI's work, the INJR work, so that IRI's global mandate is very well met by the complementary inputs of INJR. So much for the concept and what happened, but now let us see how we can progress further and take the program forward for the benefit of the participating countries. I have listed a few items here. One is the genetic cafeteria versus genetic grocery approach. By this I mean, when the national programs participate in the network, immediately there will be greater attention on the varieties which are giving higher yield. The cafeteria approach is something which is made. Just to go to a cafeteria, then okay, one day it is all right, second day it may not be suitable for your taste. Like that variety which is higher yield may be giving yield this year, but it may not reciprocate the following years. Whereas the genetic grocery approach is to see that the material as source of breeding rather than directly for giving it as a variety to the farmer. That way you can blend together the genes which are very important for the rice culture and the ecosystems, the problems that exist in the diverse cultural conditions. So that should be the focus. But initially the tendency was just pick up, okay, this is the high yielding variety, let me pick it up. But then, that would be only an ephemeral linkage with location specific cropping system trials. in every location, you would have the scientists working out the best cropping systems, because from the holistic approach for the benefit of the farmer, you have the best cropping systems are identified. If rice-based cropping systems, it is very important that the rice component of that particular cropping system should be a verified variety from on rice network trials like the India. Otherwise, what happens is we have the best trials, best varieties from the India trials. However, they may not be the best entries for fitting into the seasons that are dictated by the more profitable cropping systems. That's where it is extremely important that the cropping system trials and should not be in isolation with the variety trials. I'm just giving an example for the rice crop, but same is true with other crops also. Ultimately, it should be a holistic approach, and what are the best benefits that the farmer can get. In that context, it's extremely important. Now, we should not keep the program just to say once we worked out the types of tests in the ecosystems, then should not keep going, because the needs keep changing. There will be new parasites, and then there will be unknown problems like climate change. And so one should be cognizant of these emerging needs, and the program should be directed towards that. And that's where, once again, the facility of the network can be very profitably availed. Now, fertilizer and water use efficiency, the crop husbandry scientists, the agronomists and others, they have done their best in providing the levels of fertilizers or nutrition to be applied to the crop, and likewise the water in the water use, similarly come up with the methodologies for the more efficient use. But there should be a genetic approach to complement this. That is, the varieties themselves, are they different in their ability to effectively utilize the native fertilizers or the applied fertilizers? And the similar is the case that there is no redundancy and wastage in use of water. But before that, identify varieties which have a more capability to produce more yields under relatively, I do not say low, but with an optimal levels of water and fertilizer. So this is one suggestion that normally the fertilizers and water use, we leave it to the agronomists, and the breeders should also put in their inputs by finding the genetic variation on this particular factor. Traditional germplasm. This is what we call the gene bank, and this is extremely important if you want to utilize this, because the germplasm bank is meant for not the use of IRRI where we have the germplasm bank established, but it is meant for the entire rice growing countries. The reason why I'm pointing out on this is that Dr. T.T. Chang, my colleague, and he spent a lot of time and in collecting the rice samples from various countries. The rice gene bank and there are more than 75,000 accessions in the bag. These are all DNA fingerprinted, and then those have been also tested for the reactions to various biotic and abiotic stresses, and then the characterization on that is documented and provided. However, as I show the previous slides, there is so much of variation in terms of the pathogenic virulence and then the insect biotype variation. So from the gene bank, if someone from the national programs wants, I would like to get a genetic donor for this particular disease, you cannot confidently give it from the bank because you do not know how it reacts elsewhere. That's why I suggest, because this is something which would put a lot of burden in terms of the work time as well as the finances of the national programs. And the traditional germplasm screening, they should be specially funded. And in batches, the material can be sent out to different countries, just like the regular, the injured trials, and then get the reactions in other countries, other test sites, so that information. Instead of saying that, okay, we found in this collection this particular variety A is resistant to the green leaf hopper. And then, but that you do not know what happens in other country. But on the other hand, the next time, after you have the data, you are in a position to tell first of all, ask which location you are wanting this. And accordingly, look into the database and see and view this variation that which would be the most suitable for that location. That's why the traditional germplasm evaluation is extremely important. The last thing I'd like to point out is the seed vigor. This is, I would say, one of the uh, neglected thing on from the genetics and the breeding. Most of the traits, most in the breeding traits, I am myself a plant breeder, and I know we used to focus mostly on traits from seeding to harvest, but not the post-harvest traits. But factors like viability and vigor have tremendous influence on the seed vigor. And then you may provide to the farmer some varieties on the basis the experimentations where you have excellent conditions for storage and then find that variety is a higher yield. But God knows under what conditions the farmers store the seeds after their harvest and keep the seed for the next crop. And under the adverse conditions of humidity and temperature, the moisture in those, the quality of vigor would have dwindled to a point where you cannot expect a good crop because you give a good start to the crop if you give a good vigorous seed. But if you start with a poor seed, the crop performance is equally poor. Now I will illustrate that with the actual data we got. You would see here two varieties. On the left, you have IR 13429, and on the right, you have IR 31779. And the two here, the varieties, you see the difference that the green is the control variety without any treatment. And the red is very high yielding variety when it subjected to adverse conditions with a low seed vigor, the yields are nearly half. On the other hand, the next variety, while it has not given as much yield with the healthy seeds, it has given nearly the same seed because of its vigor. Now, how is this vigor determined there? We have two ways, but in this particular one, we call it an accelerated aging test. To subject the seed, we have accelerated aging chambers and subject the seeds to temperature of 43 degrees centigrade, humidity of 100%, and a duration of 6 days. And after you subject that, the seed you test for the simple the enzyme activity test, like the amylase activity, the peroxidase activity, the dehydrogenase activity. So these test results would show that after this moist heat treatment, what would be the seed vigor. And that's where, I am emphasizing the point that unless you build in this seed vigor genetically into the variety, it would, the farmer would not be able to realize the potential needs of the varieties. The second way, of course, is that we have the automatic seed analyzers where you pass electric current through the leachates of the seed. And we use three levels here, a part of the control, that is 20 microamps and 40 microamps and 50 microamps. And then After passing the university, then you have established 4 levels. This is, unlike the previous one, this is with a single variety. And then for the same variety, when you artificially reduce the vigor, you would see how much the yield decline is there. Because it's great importance, I spent more time on this particular reading trait, and I hope readers will take cognizance of this fact and intensify their efforts in identifying varieties that are, you cannot be getting 100%, in all these things, but definitely, in spite of whatever storage conditions he has. And in some cases, the photosensitive varieties which they keep, so they are there for a long season, and then the dormancy itself would prevent. This is the nature's provision, because the seed vigor is generally, the seed vigor is higher when there's dormancy. So of course, in tests like this, you have to first eliminate, first bring in uniform for the test of these differences. You must bring in, eliminate the environmental factor, because the seed vigor is influenced by both the pre-harvest environment as well as the genetic factors. In order to get to true genetic differences, you have to eliminate the environmental impact, and then the varietal differences can be identified with certainty. Now, to conclude, I would say the networking concept will continue to permeate virtually all aspects of agricultural research to the enduring benefits of farmers and consumers because of reduced costs, saving on time, effective use of resources, and increased research efficiency. Finally, I prefer to show my slide always this, because when you are talking of sustainable agriculture, this is extremely important, that all the resources that we inherited from our ancestors, we pass them on at the same level to your future generations. So I would say use efficiently now and save sufficiently for posterity. Thank you very much.
Sir, thank you very much for your wonderful presentation, sir. It was really, we immensely thankful to you. And if you want to, anybody wants to share their experiences, like Shastri family members, sir, now it is open for you, sir. Then our formal vote of thanks will be given by our colleague, Dr. C.H. Padmavati. Before that, any Shastri members, Shastri family members?
Seshu Garu, the presentation was it was very great. It was very simply explained, so even I could understand a lot of the concepts that you were sharing. And it looks like it's really relevant for the agricultural institutes, not just for rice, but also all the different varieties and types of crops. So thank you so much for sharing. It was very valuable.
Completely agree with what Arun was saying, that same thing, that what I got out of it is that the principles that you should use when you are researching or trying to improve a product always can use improvement. And what are the things you should look for, not just today, but for future, because we don't live in an ideal world. So how you can really look, not to shoot the moon. That's a favorite line I like. Don't shoot for the moon, but get the best you can, because that's what the real life is. It's not always shooting the moon. So I really liked it. Thank you so much for this opportunity to join the meeting.
Okay, Dr. Seshu Garu, I have a very naive question, because rarely do I have an opportunity to talk to someone as clever as yourself. I was listening to a naturalist recently, David Attenborough. You might have heard of him. He's very keen on environment and saving the world for the future, just pretty much like your last slide was showing. And I hope you can hear me. Can you hear me?
Yeah, sure.
Okay. And one of the things he was concerned about was the loss of diversity in wildlife because of the amount of land that is taken up, not just for agriculture, but also for farming, if you like, for basically to sustain the human life on Earth. And he felt that as a result of that, quite a bit of the wildlife was being diminished. And one of the examples he came up with was that in countries like Holland, they use intensive farming, like in greenhouses with intensive lights and hydroponics to grow. His example was tomatoes. And it sort of made me think that it was actually a very clever thing to intensively farm in a very small area, almost vertically, if you like, rather than taking up a lot of footprint that is historically been the case. It might be something that is not possible, but it was a question that has been in my mind for a while, and I wondered what your thoughts might be on this.
Yeah, it's hydroponics part. The first thing is that when you want to conserve the useful genes, and so that now you see there's a lot of destruction now in natural habitats with a lot of civil constructions and activities, and the sources for various traits that are economically important. Thereby, we are destroying those. Hydroponics and all that in a limited way, these could be, but the point is that if you have both in situ as well as ex situ conservation, so that you select, that these sites, what happens is when you make collections, keep them in the gene bank, they do not just stay like that. They do not change, but under the natural conditions, they keep on mutating, and you will find a lot of wild traits are shredded, and then you economically important traits. So the two-pronged approach we thought is to have both ex situ and in situ conservation. So that is my answer for that, because that would permit, a large number of the genetic resources to be maintained and available for the future generation. Of course, we have near the North Pole in Svalbard, under very cold conditions, the seeds from various crops have been stored there and not destroyed by earthquakes or other activities. I do not know whether I answered your question on that, because I did not dwell on the hydroponics, because I'm only thinking of the mass retention of the genes. And in that, while that is to grow those under the controlled conditions and all that, would give some empirical information. But for it to be translated for practical thing, that we need to have these approaches.
Yeah. Thanks for your info. Thank you. I'll continue to think about it.
That's a good question.
Wants to say anything? If not, I request our colleague, IIRR colleague, Dr. C.H. Padmavati, to say vote of thanks.
Yeah. Thank you, Mahinder. Can you hear me? Yeah. And thank you, Dr. Mahinder. And namaskar mandi andarki. Namaste. I'm C.H. Padmavati, working as principal scientist in entomology department at Indian Institute of Rice Research. So it's a great pleasure to propose the vote of thanks. I thank today's guest, Dr. Seshu Garu, for readily accepting and delivering the Dr. HVS Shastry Memorial Lecture today, sir. I thank you very much. And we cannot think any other person than experience of working with two largest networks, I can say. One is national, India's largest network, that is ICAR for 10 years, and then followed by world's largest network, India, for 20 years, sir. So thank you for sharing your vast experiences of working with India and also ICAR. And I thank you for showing the rare photographs, which really we, at IIRR we don't have such rare photographs. I thank you very much for sharing those, showing those photographs also. And I thank your family members also, sir, who are present here. I can see Padma Garu and Samir Garu. Thank you very much for delivering a very, very useful lectures for today, sir. And I thank the family members of Dr. HVS Shastry Garu, Arun Garu, Roja and Raja Garu. Thank you very much for being present here. And I, as you said, yes, I think COVID has given us an opportunity to see and meet like this. And I also thank our director of Indian Institute of Rice Research, who is also the president of Society for Advancement of Rice Research, SAR. I thank him for conducting this and also giving us this opportunity of meeting you all. And I thank Secretary of SAR, Dr. Mahendra Kumar also, and all the members of SAR who are present here. And I thank each and everyone who has helped us in hosting this and also in conducting this. We will be again, as Dr. Mahendra said, we will be retesting this on 15th December 2020, which is our actually IIRR Foundation Day. I also thank Dr. Sailaja and her team links and these arrangements. Thank you each and everyone who has helped us in conducting this memorial lecture. Thank you.
Thank you, sir. Thank you, one and all. Seshu garu, our special thanks and namaskars from India. Thank you.
Thank you. Thank you. All the best.
Thank you.
Thank you. Thank you.
Thank you, sir.
Hope I first of all wish all of you a very, very happy New Year and stay safe from this virus. And I know the vaccine is there, and now I hope the 21 will be a better year. And as Roja said, you know, that we had this way an opportunity to see each other, but I hope and wish that the actual program which you have on Tuesday very high success. And is it possible for you to send us the recorded document of that?
Yeah.
We will also send you links, sir,
Yes.
as well as a recorded one also we can share with you. We will also send the link. If you can able to see live program, you can see. Otherwise, we also send the link, recorded one afterwards.
Bye all the best.
Thank
Thank you, sir.
you. Thank you very much.
Happy New Year, sir.
Thank
Happy
you.
New Year.
Roja and Arun, thanks.
Thank
Thank
you.
you, Seshu garu.
Okay. Glad to see you all.
It's lovely to see you.
Likewise.
Shailaja, now you can discontinue the meeting. Thank you, sir. Thank you all.
Bye. Bye.
Thank you.